And then next up we have, do you prefer Alex or Alexander?
Alex.
Alex. We've got Alex Wade, who's going to be talking to us about addressing the challenge of drug discovery with machine learning and exascale computing. Go ahead, take it away, Alex.
Okay, good morning, everyone. My name's Alex, and I work in the group of Professor Peter Coveney on the application of drug discovery methods to high-performance computing environments. In this talk, I'll kind of discuss some of the machine learning approaches we're looking at, and then the challenges and opportunities we anticipate with scaling these up to larger class machines. So before I begin, I'd like to thank our collaborators and sponsors on these projects, and I'd like to give a special thanks to the other members of Peter's group, Shanzu, Agastya, and Matt, who have actually performed a lot of the work that I'm going to present here. As a summary for what I'm going to speak about, first, I'll kind of give a very high level but brief introduction into what kind of quantities we're calculating in these drug discovery campaigns, and these are kind of binding affinities or binding free energies, what these are and why they're important. Then I'll speak about our more traditional physics-based approaches, and these are atomistic simulations of the protein and the ligand. And then I'll follow this with discussion of our machine learning approaches, speaking about specifically a project we were involved with the open source malaria consortium. And this was basically a project, an open source project looking to find novel anti-malarial compounds. And then I'll conclude with a discussion of our new hybrid machine learning and physics-based methods. So in this approach, we're kind of trying to trade off the accuracy of the physics-based methods with the speed that we can get from the machine learning methods. So in a drug design campaign, we're typically looking for a small molecule which binds non-covalently to a protein, and this protein might be involved in the mechanism of action of some disease. And by binding to it, we hope to maybe inhibit it or modulate the behavior of this protein somehow and treat the disease ultimately. So a gross oversimplification then is that this binding affinity can be used as a rough proxy for the effectiveness of the drug. One of the problems with drug design is that the space of all drugs is very, very large. So if we're looking for a particular small molecule which binds to a particular protein with some set of properties, this is a very hard problem to solve, and it's basically a needle in a haystack type problem. Drug discovery campaigns have kind of methodologies to deal with this, like high-throughput screening or virtual screening, but high-throughput screening is experimental, and the costs for this can rack up quickly. Virtual screening, if it relies on more simple methods like molecular docking or pharmacophore modeling, misses some of the essential physics of the problem. So ideally, we'd like to include things like protein flexibility or the entropy of binding naturally in the calculation. And this is where we can use alchemical free energy methods, which rely on molecular dynamics. So molecular dynamics is a common simulation technique. We use it a lot in our group. What's interesting about molecular dynamics is, despite the fact that kind of the equations of motion are deterministic, we see chaotic behavior in these simulations. And this is borne out in the free energy calculations that we're doing, that instead of just seeing a single measurement, we get a distribution of results. What I'm speaking about here is basically an aleatoric error, which we can control for with replica simulations, but we also have a parametric uncertainty. So Peter's group has developed this suite of tools called the Vecma Toolkit for kind of automating the calculation of parametric uncertainties. We've applied this Vecma Toolkit to a popular molecular dynamics package called NAMD. And for a free energy calculation in NAMD, you might have some 175 free parameters. So from this work, this was whittled down to 6 parameters, which are ultimately significant for the uncertainty in the binding free energy calculation. And these were temperature, box size, and barostat parameters. Generally, overall, the result here was that the uncertainty in the input parameters was damped in the uncertainty of the free energy calculations. So this is a good result for the robustness of these kind of methods. Our specific flavor of kind of molecular dynamics and alchemical methods as applied to binding free energy calculations is called TIES, which stands for thermodynamic integration with enhanced sampling. And This is a method which includes the aleatoric and parametric uncertainty quantification that I was speaking about in the previous slides. As such, it's kind of an expensive method and it uses a lot of replica simulations. So typically one binding free energy calculation in ties might use 65 GPUs for around 2 hours or so. But with kind of this uncertainty quantification, we're very happy with kind of how actionable these results are and reproducible. And we've tested this with multiple molecular dynamics engines, many proteins, hundreds of ligands, with different free energy estimators and enhanced sampling and without enhanced sampling. And we get very good agreement with experiment across the board. Another method that we use is called ESMAX, and now this is a continuant solvent method using MMPBSA. What's important to know about this method is that generally it's cheaper than ties. So it might use 25 GPUs now for 1 to 2 hours, depending on the size of the protein. This reduced computational cost comes at the price of accuracy. So typically we might only use ASMEX for the ranking of compounds. And these are some of the results that we get from ties and ESMAX calculations. So what we can see here is that the distribution of the delta G, or these binding free energies, is not necessarily Gaussianly distributed. So this comes back to what I was saying before about the need to run replica simulations. Like this is doubly true if these are not nice Gaussian distributions. And we see this across the board with generally kind of skewed and distributions with kurtosis for these kind of calculations. And this is not something that we just observe computationally. These are results taken for experimental binding for energy measures from GSK for an SMYD3 inhibitor for 4 ligands, A, B, C, and D. And you can see here as well that these binding for energies are not necessarily having a nice Gaussian distribution. So these are the physics-based methods. They're generally expensive and we have to use replicas to quantify the uncertainty. They can be very accurate, but how can we accelerate this now with molecular dynamic, with machine learning approaches? So this was an interesting piece of machine learning work that I was a part of, and this is the open source malaria project. So this was an open project where entrants from academia and industry put forward kind of discriminating methods to try and identify or predict active compounds in this existing series of antimalarial compounds based on this S4 core here. So the reason machine learning methods are kind of very relevant for this test case is that there's no definitive mechanism of action for this series of compounds. So although it's speculated that these compounds work by acting on an essential ion pump on the surface of the parasite, we don't have a target, we don't have a crystal structure, so we can't use these molecular dynamics approaches. The structure of this competition was that 10 entrants began at the beginning, again, coming from industry and academia. They developed their discriminating model to predict on an unseen data set, and they used a wide variety of methods: deep learning, homology modeling, and linear regression. Overall, I guess the general trend here is that these networks can be lightweight. So they're not using millions of parameters, maybe a few thousand, and so they can predict very quickly. And this allows us to explore much larger regions of chemical space than would be possible with physics-based methods. From the most precise discriminating methods, 4 teams were selected to move to the second round. And now we're doing a prospective study where we're actually generating compounds or predictions for new actives in this series. So again, we've got 4 teams participating here. So we've got a variety of methods being applied, from recurrent neural networks to medicinal chemistry expansions and human collective intelligence type approaches. Although some of these methods are directed, like the recurrent neural network, the general overall idea is that this is an exercise in producing as many molecules as you can and then throwing these at the discriminators. And then we're free to take this lazy approach again because the discriminators are so quick to give us a proxy for the binding free energy. From the 4 teams, 6 molecules were actually synthesized, and half of these showed some activity. The one in the bottom left was the most active one, which is this tert-butyl pendant motif. This molecule was actually an interesting data point because it was kind of rejected by the human team that were familiar with this series as probably being inactive, but it was active in the end. So this was a valuable data point for their ongoing work, trying to determine the mechanism of action and find QSAR relationships for this compound series. So what lessons did we learn from this project? Well, everything I've shown up until now was for this series 4 core, but there was another area in which we could make kind of suggestions or for new compounds, and this was for structurally diverse compounds. So this is a much harder problem, and these are my suggestions for the structurally diverse compounds along the bottom. We were the only team that was naive enough to try and make predictions here using our methods. So I'm happy, since these are my predictions, to critique these very harshly in that they're not active at all and definitely won't be. So yeah, but this is a known problem of these kind of machine learning methods where they're not going to extrapolate far away from your training data. And so this is kind of an expected result in a way. But what if we want to do this? What if we want to find, maybe do scaffold hopping or to be robust against this kind of mistake? And then this is where kind of hybrid machine learning and physics-based methods can come in. So when we talk about hybrid physics-based and machine learning approaches, we kind of mean this multiscale modeling approach where in the beginning, you might start with a very large library of compounds. You filter this first with your cheapest machine learning methods, and then you pass this on to your physics-based methodologies with the hope that you miss fewer opportunities in the beginning, but what you have at the end has the uncertainties quantified on it and is kind of an actionable result. Our specific flavor of this multi-scale modeling type approach is called Impeccable, and this starts with a machine learning surrogate model for docking. This is designed to be very quick and just give you a proxy for the binding free energy. From this surrogate model, the best compounds are selected and passed on to more traditional docking, which gives us a 3D structure, a complex, which we can then simulate with the physics-based methods. We would first start with simulations using ESMAX, which is the cheaper of our methods, and then finish with TIES, which is the most expensive method. The results from the ESMAX and the TIES simulations can be passed back into the machine learning surrogate such that it learns what it correctly and incorrectly predicted as active. So this is kind of not just a funnel anymore, it's more of an iterative cycle where you hope that over time you're kind of learning the interesting and reliable areas of chemical space. We've applied this workflow at scale on numerous different HPC machines, Frontera at TAC, Summit at the Oak Ridge National Lab. And we found for scaling up to these machines, it was very useful to use a middleware solution. So here we're using something called Radical Cyber Tools, and this allows you to automate and orchestrate your simulations to schedule them easily in this kind of complex workflow. But it also allows you to target heterogeneous resources. So you can run part of your simulation on CPU and part on GPU, and this avoids wasted cycles and lets you use a bigger fraction of the machine. So this is kind of validation that we've done on pre-exascale machines, but how would this scale up to use even more resources? So large parts of this workflow are what we call pattern 3, or this is the same as kind of embarrassingly parallel. So there's no communications between, say, the individual molecular dynamic simulations in TIES or evaluations of a ligand with the machine learning surrogate. And so we're free to parallelize these much easier over large resources than the general case where there's communication. So with our kind of middleware approach and the validation that we've done on pre-exascale machines, we're confident that we should be able to scale these up to exascale class machines. So in terms of the pre-exascale validation that we've done, we've run large block operations on machines like SuperMilk NG, which is a CPU-based machine at the Leibniz Supercomputing Center. And we've run on up to 6,000 nodes, running these binding affinity calculations with TIES and ESMAX. Likewise, we've run similar block operations on Summit, and this is where Impeccable was run, using up to the whole machine to kind of run this workflow. Interestingly, on Summit, these middleware tools were not just kind of helpful to scale up, but actually imperative to be used. We found that we could very quickly overwhelm the batch nodes, which normally schedule the jobs without some kind of middleware solution, and bring the whole machine down. So these were really helpful to use, these middleware tools on Summit. And with that, I think I'm finished, and I'd like to open it up to questions.
Great, thank you for the talk. As someone who also has run workflows, this is a big feat. So this is really impressive work. Okay, questions for Alex. All right, we've got a few. This one first, and then I'll come back to you, and then Oles, you're after that.
Yeah, thanks, Alex. Great talk. For the data you presented on the non-Gaussian distribution of binding affinities, yeah, that one. Have you done experiment like allocating more sampling and rerunning this data set? What happens to the distribution if you put more compute effort in getting fringe estimates for individual replicates?
We haven't actually looked at how this non-Gaussian nature changes with respect to the number of windows and the amount of sampling, but this is something that we do with respect to the convergence of the simulation and stuff like this. But it would be an interesting idea to see if it varied with the number of alchemical windows. I don't think it would. Yeah.
Okay, yeah, thanks.
Hi, Alex. Thank you for an amazing talk. So I'm very curious about your binding affinity calculator. So from the ensemble of each MD engine, you get the distribution, or you get which part of the trajectory is the best?
Can you say the first part again? I missed it, sorry.
And the binding affinity calculator, I'm interested to know how you calculate that. From the MD engines, you get as a result the ensembles of the trajectory, or you get the distribution?
So we would run, say, 5 replicas of the solvent simulation and of the complex simulation, and then you would average these and then add those together in the thermodynamic cycle. So, yeah.
Sorry, Rafa.
Quick question. In your Impeccable workflow, I guess, yeah, can you comment how successful you were to pass in the computed free energies to the surrogate model, and how easily you can learn
So this is
delta
not,
G's?
This is not a workflow that I developed or I'm expert on. This is something that other people are working on in Peter's group, but I couldn't really comment intelligently on how easy it was to get it to learn from the physics-based results. So.
Okay, I'll follow up.
All right, do we have any other questions for Alex? Okay, and while we're taking that question, I'll ask the next speaker to please start setting up. And sorry, I actually lost the person who had the question.
So maybe this is a little bit ill-posed because it's also about Impeccable. But I was sort of wondering if you had thought about synthesizability when you implemented this workflow, because obviously it's a large compound sort of screening, but oftentimes the results that you get are impractical for many actual applications.
Yeah, so this is a really good question. This is something I can comment on. So particularly more from the OSM open source malaria type work. So yeah, synthesizability is a huge problem. Toxicity is a huge problem. Ideally, you might want to kind of include some other machine learning models in this early part of the Impeccable workflow, such that you're screening for these things that you're interested in. This is, something that would be kind of further work. And in the open source malaria stuff, this is all done by hand. So like massive human intervention to select stuff out that looks obviously wrong and broken. So yeah, I think it's an open problem.